Every moment of every day, every single cell in your body is engaged in this complex high-stakes communication. Should I divide? Should I die? Should I migrate? This constant political landscape, this coordination of trillions of specialized units, it's governed by a complex and often, well, astonishing language of molecular signals.
It's true. And that language is the focus of our deep dive today. Cellular activation. We're looking at the core mechanism of intercellular communication. Our mission is to take the dense source material, the nuts and bolts of molecular biology, and really understand the mechanics of how a cell interprets that external cacophony of signals. And translates it into a rational, coordinated internal output.
Yeah, and the stakes here aren't just academic. They are literally life and death. Our sources really stress that this highly coordinated signaling assures healthy tissue organization. Whether you're, say, rapidly building a scar after trauma, marshaling defenses against an infection, or just maintaining daily turnover, it's fundamental.
Absolutely. Yeah. The problem arises the moment you lose that highly sophisticated social control. Yeah. When communication breaks down, you get catastrophic, unregulated consequences. That could be the unchecked growth we see in cancer, or it could be a detrimental, uncontrolled, systemic response, like immune shock.
Wow.
The whole process, from beginning to end, requires 3 core elements. You need a signal, a specific receptor to receive it, and then the internal wiring to process it.
Okay, let's unpack this, starting with the vocabulary itself.
Fair.
The signals.
Right.
So what exactly is doing the talking? Our source material breaks the input down into 4 fundamental categories, which seem to range from immediate physical proximity to universal broadcasting.
That's a good way to put it. The first 2 are about immediate awareness, sort of the cellular neighborhood watch, if you will. The cell has an innate capacity to sense danger and pathogens, signs of damaged cells or microbial invaders. These inputs trigger immediate defensive reactions. Pretty crucial.
Makes sense. And then we move to physical contact, which has 2 forms. The first is direct cell-cell contacts, often mediated by adhesion molecules, but also, you mentioned crucially, by gap junctions.
Gap junctions are fascinating. They are literally hydrophilic channels, these things called connexons, that span 2 membranes, allowing the immediate movement of tiny molecules. We're talking about small ions like calcium metabolites or critical second messenger molecules such as cyclic AMP, GMAP, moving directly between adjacent cells.
Okay, but why is that mechanism superior for tissue coordination? Why not just, secrete a molecule?
Speed and synchronization. It enables instantaneous coordination. If you need a sheet of cells, say smooth muscle cells, to contract simultaneously, you need that immediate direct flow of electrical and chemical signals. It's basically the highest bandwidth, lowest latency communication available to a cell. Really direct.
Got it. High speed, low drag.
Okay.
The third category of input is structural support. Cell-ECM contacts, ECM being the extracellular matrix. These are usually mediated through specialized molecules called integrins.
Integrins are essential because they act as mechanical sensors. They tell the cell where it is anchored, what the stiffness of the surrounding tissue is, and what tension it's under. This constant feedback loop between the cell and its environment is what prevents cells from, say, moving where they shouldn't. It's a concept that's incredibly important when we later discuss pathological conditions like inflammation and metastasis.
Right, holding things in place. And finally, the universal broadcasting system. Secreted molecules. This is the most famous group, I guess. Growth factors, cytokines, the inflammatory mediators, and hormones, the endocrine long-distance runners.
Exactly. And that reach, the spatial relationship, is how we classify the delivery system. If the molecule only affects its immediate neighbors because it's rapidly degraded or trapped by nearby matrix components, that is paracrine signaling.
Like shouting across the cubicle. I like that analogy.
Yeah, pretty much.
But sometimes the cell needs to signal itself. That's autocrine signaling.
Right. This is often employed during development to maybe amplify an initial weak signal or to synchronize groups of cells in a positive feedback loop, kind of like boosting its own message.
Okay.
Then you have the rapid-fire, highly specific delivery system. Synaptic signaling. This is used by activated neurons secreting neurotransmitters across tiny specialized junctions. This is incredibly fast, allowing for things like muscle contraction or thought processing. Very targeted, very quick.
And for the grand, slow-moving, body-wide broadcasts like the regulatory signals affecting metabolism or temperature, we have endocrine signaling.
That's the one. The mediator, often a hormone, is released directly into the bloodstream to act on target cells far away. Think insulin regulating blood sugar across the entire body.
So the signal is delivered, the cell is bathed in this molecular chatter. The next critical step is interpretation. That brings us to receptors, the cellular receivers.
Exactly. And what's really key here is the precision required.
Yeah, the source mentioned that. The signaling molecules, the ligands, must bind to their specific receptor proteins with high affinity and incredible specificity. It sounds like a lock and key, but maybe even more precise.
It really is. The receptors are the primary transducers. They take that chemical message from the outside and change their conformation, their shape, to launch an internal cascade. We categorize them mainly into two groups, the insiders and the gatekeepers.
Okay, the insiders, these are the intracellular receptors. They're usually floating around in the cytoplasm or the nucleus.
That's right. These receptors can only be activated by specific types of ligands molecules, like vitamin D, thyroid, or steroid hormones. These are special because they're small and nonpolar enough to easily slip right through the lipid plasma membrane without any help. Once inside, they bind their receptor, which then often acts directly as a nuclear hormone receptor, fundamentally changing gene expression. Goes right to the source code, so to speak.
And there's that elegant outlier you mentioned, nitric oxide, NO. It's small and nonpolar too, right?
Yes. It just diffuses from an endothelial cell into the adjacent smooth muscle cell. It doesn't use a nuclear receptor in the typical way. Instead, it activates a cytoplasmic enzyme, guanylyl cyclase, generating cGMP, which causes the smooth muscle to relax. That single mechanism is fundamentally what regulates your vasomotor tone, changing blood pressure instantly.
Wow. It's like the cellular equivalent of using a tiny puff of gas to throw a massive structural switch.
Exactly. Now contrast that with the gatekeepers, cell surface receptors. These are almost always transmembrane proteins, sticking through the membrane with an external binding domain. They handle all the signals that cannot cross the membrane, most peptides, growth factors, cytokines, that kind of thing.
Right. And ligand binding here results in 4 distinct actions, according to the source. They can open ion channels.
Leading to instant electrical changes like in a synapse.
They can activate an associated G protein.
Which we'll definitely get into. Big category.
They can activate an enzyme, often a tyrosine kinase.
Also very common, especially for growth signals.
And the 4th critical action. They can trigger a proteolytic event or a major conformational change that releases a latent transcription factor.
Yes, this is common in those critical developmental pathways, like Notch or WANT. And the sources point out that defects in these cell surface receptor signals are often central to malignancy. So, really important pathways.
Okay, here's where it gets really interesting for me. We have the signal, the receptor has captured it. How does the receptor translate that external hello into a durable internal action? This is the signal transduction pathway, the internal wiring.
Right, the wiring.
Yeah.
And the universal currency here across nearly all pathways is phosphorylation, or its reversal, dephosphorylation. It's the simple act of adding or removing a charged phosphate residue to a target molecule. This tiny chemical modification acts like an on-off switch.
An on-off switch.
Exactly. It dictates whether an enzyme is active or inactive, or whether a transcription factor can get into the nucleus. It's the fundamental language of internal cellular decision-making.
Okay, so let's start with the kinase receptors, the ones that do the phosphorylating. The receptor tyrosine kinases, RTKs, like the receptors for insulin, EGF, or PDGF, these seem highly organized.
They are. They're self-contained machinery. When the ligand binds outside, it forces 2 RTK subunits to physically cross-link or dimerize, basically pair up. This physical association instantly activates their intrinsic kinase domain, which is located on the cytoplasmic tail, inside the cell.
So they phosphorylate themselves.
They phosphorylate each other. It's a process called autophosphorylation, and suddenly they are fully active signaling hubs ready to phosphorylate other things.
Okay, but then you have the non-receptor tyrosine kinases. The source mentions receptors for many immune molecules, cytokine receptors, even integrins lack that built-in kinase activity. They're like an antenna without an engine.
Good analogy. They have to rely on associated intracellular contractors, like the important SARC-C family kinases, to do the phosphorylation work for them. What's crucial here is how they link up. These SRC kinases use specialized modular connector domains. You might hear about SH2 and SH3 domains to grab onto the activated receptor complex and aggregate other enzymes. They build a platform for signaling.
Promoting complex protein protein interactions, like little organizers.
Precisely.
Which brings us to arguably the most famous family of receptors, the G protein-coupled receptors, or GPCRs. These are the serpentine receptors, right? Snaking back and forth across the membrane 7 times.
That's them. There are over 1,500 identified, maybe more now. They regulate everything from vision and smell to hormone responses. Huge targets for medicine, too.
So how do they work? The source mentioned a molecular game of musical chairs.
Yeah, it's kind of like that. Ligand binding outside causes the GPCR to change conformation. This change allows it to interact with an associated G protein on the inside. This interaction forces the G protein to eject its low-energy guanosine diphosphate, GDP, and rapidly bind the high-energy guanosine triphosphate, GDP. It's like trading a dead battery for a charged one.
And the moment that G protein is GTP-bound, it's active, switched on.
Exactly. That activated GTP-bound subunit then breaks away and initiates the downstream pathway. This often involves generating critical second messenger signals like CAMBP or IP3. And IP3 famously triggers this massive and rapid calcium release from internal stores, the ER, leading to a huge internal response. Very dramatic.
Wow. Okay, we also need to touch on those developmental pathways, Notch and Went, because the source says they kind of skip the complex phosphorylation cascades and rely more on physical or proteolytic change.
Yeah, they're a bit different. The Notch pathway is maybe the most direct example of signal transduction you can imagine. Ligand binding outside causes the receptor to literally be cleaved, cut, by a specialized enzyme. The resulting cytoplasmic piece, called intracellular Notch, doesn't wait for phosphorylation. It migrates directly into the nucleus to physically form a transcription complex.
Immediate, clean, direct, no messing around.
Pretty much. And the one-frizzle pathway is all about controlled destruction. Normally, the cell constantly has this elaborate destruction complex working overtime to identify and tag a protein called beta-catenin for continuous degradation, just chewing it up.
Okay.
But when one signaling is activated, it disrupts that entire destruction complex. This stabilizes beta-catenin, allowing it to build up, enter the nucleus, and switch on target genes.
So it's a system designed to keep the cell quiet unless a specific, powerful external signal demands growth or change. It's intrinsically off.
Exactly. It's inherently restricted.
Okay, so now if we try to connect all these individual mechanisms to the bigger picture, it becomes clear that cellular signaling is rarely just a simple linear A to B pathway.
Oh, absolutely not. It is a highly complex interconnected network.
Yeah.
And understanding this holistic view is really the domain of systems biology.
Right. Our sources use a great computational analogy that helps visualize this complexity. They say the protein-protein complexes, those spots where the cytokineses and RTKs gather, are the nodes. And the biochemical events feeding into or emanating from these nodes are the hubs.
Yes. That's a critical insight. It means you don't just have one pathway running in isolation, you have massive crosstalk. Signals from one receptor can influence the output of another. Those adapter proteins we mentioned, the molecular scaffolds using SH2 or SH3 domains, are literally promoting the assembly of these complexes. They act as central command centers, where multiple signals converge and get integrated.
This shift in perspective explains why maybe single-target drugs often fail or have side effects, right? We aren't dealing with a simple on-off switch. We're dealing with a fully networked city. Turning off one light might just cause 3 others to get brighter or dimmer unexpectedly.
Precisely. It's much more interconnected than simple diagrams suggest. And this complex signaling ultimately funnels down to produce a durable effect, which is usually modulating gene transcription via transcription factors.
Right, the ultimate internal messengers, proteins like MYC, JUN, or the famous TP53 that regulates cell division and survival.
Exactly. They move from the cytoplasm into the nucleus, find specific DNA sequences, either promoters right near the gene or sometimes distant long-range regulatory elements called enhancers, and then they recruit the necessary co-activators and RNA polymerase to start making new proteins.
And their activity, as you said, is heavily modulated by phosphorylation, ensuring the signal is strong and appropriate enough to commit the cell to a whole new program.
That's the core idea.
So we've tracked the signal from outside the cell all the way into the nucleus. What does this massive signaling apparatus actually mean for cell growth and survival? It sounds like we must talk about the powerhouses, growth factors.
Yes, absolutely. Growth factors are like the molecular green light for proliferation. But they do more than just that. They perform several critical tasks, promoting entry into the cell cycle, relieving blocks that might prevent cell cycle progression, importantly, preventing programmed cell death or apoptosis.
Keeping the cell alive.
Right, and enhancing the synthesis of all the components, nucleic acids, proteins, lipids that are needed for division, building blocks.
But importantly, the source notes they aren't just for division. They also regulate non-growth activities like cell migration and differentiation.
Correct. Let's look at a few quick examples from the source, table 1.1, I think, to appreciate their specialized roles.
Okay. EGF and PGF-alpha are known to be strong mitogens, meaning they promote mitosis or division, for a wide range of epithelial cells and fibroblasts, kind of general accelerators.
Pretty much, yeah. Then you have HEF, or scatter factor. It not only enhances proliferation, particularly for liver cells, hepatocytes, but crucially, it dramatically increases cell motility.
It tells the cell to move and divide.
Exactly. Scatter factor is a good name for it.
And the essential vascular signal is VEGF, vascular endothelial growth factor. It stimulates endothelial cell proliferation and increases vascular permeability. That sounds like the foundational signal needed for angiogenesis, growing new blood vessels.
It absolutely is. Critical for development, wound healing, and unfortunately, tumor growth.
Okay, then PDGF, platelet-derived growth factor. The source says it's chemotactic, meaning it calls in inflammatory cells like neutrophils and fibroblasts.
Right, it recruits help. And it's also a key stimulator of extracellular matrix synthesis. So very important for wound healing and tissue repair.
And finally, the highly contextual TGF-beta. While it stimulates ECM synthesis, also a wound healing function, it's also a powerful suppressor of acute inflammation. That seems counterintuitive.
It does, but it makes sense. It acts as a necessary brake, a modulator that turns off the initial inflammatory response once the healing phase needs to begin. It prevents overshoot.
I see. Balancing act.
Always a balancing act in biology.
Okay, so the pathological connection here really brings the entire discussion into focus. Uncontrolled proliferation results when growth factor activity is dysregulated, or when any part of that incredibly complex signaling pathway we just discussed becomes permanently constitutively active, like the on switch is stuck.
Exactly. Acting as if the growth signal is always present, even when it's not.
That's why so many of these genes, the ones encoding receptors, kinases, G proteins, transcription factors, are classified as proto-oncogenes. They're essential for normal growth.
Absolutely vital.
But a gain-of-function mutation, just one bad change, instantly converts them into a dangerous oncogene that drives unfettered cell division.
And we see this played out constantly in malignancy, in cancer. For instance, common lung, breast, and head and neck cancers often show mutations or amplification, meaning too many copies, of the EGFR1 gene, also called ERBB1, or consider the overexpression of ERBB2, better known as HR2, in a subset of breast cancers.
Right. The good news, though, is that because we understand this signaling, these specific hyperactive receptors are now prime targets for modern cancer treatments, right? Targeted therapies.
Absolutely. That understanding is driving personalized medicine.
The elegance and the sophistication of this whole system are truly astounding when you lay it all out. Cellular activation is this meticulously layered system, from the immediacy of a gap junction signal to the vast redundant signaling network managed by these hubs and nodes.
It really is.
Yeah.
And the detailed study of the system highlights why even seemingly minor errors can be catastrophic. Think about a mutation in just a small peripheral signaling molecule, like the RAS protein. If that mutation merely delays the hydrolysis of GTP, just slows down turning off that high-energy switch, that small delay translates into perpetually augmented proliferative signaling.
Just a slight delay.
Can ultimately lead to serious unstoppable disease. The timing and regulation are that critical.
And thinking back to the complexity of something like the Wnt pathway, where the cell literally has to constantly work to destroy beta-catenin unless a specific external signal actively stabilizes it, what does this relentless need for external input, this constant requirement for permission, suggest about the fundamental fragility and, I guess, dependence of even the most resilient cell?
That's a profound question.
That dependency, the constant reliance on the environment to permit survival and action, well, that's something for you to mull on.
That was our deep dive into the intricate world of cellular activation.